we can already get questions through the chat, and we'll answer them at the end of the webinar. So for now, I'd like to introduce our next speaker, James Otvos. Jim? All right. Hi. So, Dr. Otvos received a PhD in comparative biochemistry from the University of California, Berkeley, and a postdoctoral training in molecular biophysics at Yale University. He spent the following 20 years in academia, first on the chemistry faculty of the University of Wisconsin-Milwaukee, and then as professor of biochemistry at North Carolina State University, during which time he developed new technology for measuring lipoprotein particles using nuclear magnetic resonance spectroscopy. He then founded LipoScience, a company devoted to clinical translation of NMR diagnostic testing, and served on the board of directors and as chief scientific officer, overseeing analytical development and clinical research. Following LabCorp's acquisition of LipoScience in 2014, Dr. Otvos continued to oversee collaborative research to develop novel NMR metabolomic assay addressing mainly cardiovascular, diabetes, inflammatory, and mortality risk. He retired from LabCorp in 2022. Dr. Otvos maintains academic affiliations with the University of North Carolina at Chapel Hill and the National Heart, Lung, and Blood Institute of the NIH. He has co-authored over 250 scientific publications. He is a named inventor on over 40 patents and has received several research awards from the American Association of Clinical Chemistry, as well as the Christopher Columbus Foundation Discover Magazine Award for Technological Innovation. So, Jim, we're looking forward to your presentation.
Okay, very good. Thanks for the introduction, Ellie. I'm going to plunge right in. We've heard a lot about mass spec today, and the advantages of NMR have been mentioned, and they are substantial in terms of the ease of measurement and the efficiency. So that's the good news. NMR is the best modality for clinical translation of metabolomics, so long as the metabolites are present at high enough concentration, namely roughly greater than 10 micromolar. The bad news, of course, is that almost all of the metabolome consists of metabolites of lower concentration, so that's in the realm of mass spec. So really what I want to talk about today is the good news, what one can glean from an NMR spectrum of blood plasma. The NMR spectrum itself can be considered to be untargeted because you get the whole spectrum whether you want to use it all or not. What is challenging, or certainly more challenging, is the quantification of individual metabolites that give rise to observable NMR signals in the spectrum. And that challenge is due to the high degree of overlap with other signals. But it is possible to get a lot of information from a very short, less than 1 minute, so-called blood scan. These are some of the metabolites that can be extracted that I'll talk about a little bit. And then in the brief time I have available, I'll just mention some primary clinical applications that I think really should drive the introduction of NMR into routine clinical testing. So the road from then to now, it really extends back to 1986, so more than 35 years ago. I'm sure some of you weren't born then, but that's when I first got interested in this from an academic standpoint. And then this became clinically translated by a company called LipoScience and subsequently LabCorp. But what I say at the top is that broad clinical translation is still a work in progress because you probably don't have NMR analyzers in your own laboratory, and that's really what still remains to be accomplished. So I'm going to walk you through quickly each of these four phases in the history of then to now. So it began with a paper in the New England Journal of Medicine in 1986 saying that you could diagnose cancer in a very simple way by looking at a proton NMR spectrum of blood plasma. And that created a tremendous amount of interest in the NMR community, including myself. But unfortunately, many other people could not reproduce the phenomenology. So a paper published 4 years later in the New England Journal basically put an end to the idea that NMR could serve this purpose. But in the meantime, it caused us and others to look more closely at what was the underlying phenomenology. And it turned out that the signal that was the so-called cancer diagnostic was a signal from the lipoproteins in blood. And so in the early 90s, we published a couple papers showing that the NMR signal from the lipids in smaller lipoprotein particles versus larger showed up further to the right for the smaller particles, further to the left in the larger particles. And this made it possible to take a plasma spectrum, which is the mixture, and with an understanding of what the underlying signals look like from the contributing lipoproteins, one could quantify LDL, HDL, VLDL, and its subspecies. And so that was patented in 1990 at the very beginning of this trail. So what the process is of taking the NMR spectrum and converting it to quantitative information is dealing with this spectral overlap, which is severe in the case of the lipoprotein subspecies, as you can see here. And so this is, we use a deconvolution approach. In this particular picture, you also see signals from branched chain amino acids. So these are all quantified simultaneously. And this has all been well worked out and has led to the ability to report concentrations of many lipoprotein subclasses, as well as small molecule metabolites and some multi-markers as listed here. I don't expect you to read any of these things, but all of these things that are reported and able to be reported from a single NMR scan are well analytically and clinically validated. Just one quick example of the need for deconvolution is this so-called glyc-A signal, which is an inflammation marker that I'll talk about a little bit later. And you can see here on the left that this fairly sharp signal shows up on the left side of a broad shoulder that comes from lipoprotein subspecies. And you can see by these four spectra of four different people with different triglyceride levels that the overlapping signal looks very, very different depending on the individual. And this really needs to be dealt with by a complex deconvolution model that makes sure that glyc-A does not include in its quantification lipoprotein information or protein information. So I mention this just because some other folks are reporting glyc-A by NMR whose concentrations are about 3 times larger than the ones we report. And that's because they simply integrate the entire signal and don't really worry about the overlap of the lipoprotein signals. So back to sort of the second phase of this process of commercialization or translation of NMR diagnostic testing. So this was conducted by a company called LipoScience that was established for this purpose. And what it was a lab testing business for its first several years, and then it transitioned into an IVD business with the motivation to make the NMR analyzers available to any laboratory, and that was successful to an extent. But then the company was acquired by a lab company, a lab testing company, LabCorp, and so the IVD business was sort of discontinued. So I'm going to walk through some of these sort of 3 phases of the LipoScience history very quickly. So this is what the NMR lipoprofile test looked like, a 2-page color report that was used for patient care. I'm not going to go into any of the details, but LDL particle number was the main value proposition, but also HDL particles and then insulin resistance index that I'll talk about later. So the practical challenge in trying to start a company devoted to NMR testing was that NMR spectrometers generally look like this. They don't look anything like a clinical analyzer. They have a big footprint. They're not really optimized for efficiently conducting one experiment. They're engineered to be multifunctional. And so what had to be done was to modify research NMR spectrometers into the first NMR clinical analyzers. And so here you see a room at LipoScience with about 15 of these modified Bruker 400 megahertz NMR spectrometers with these Gilson liquid handling systems sitting on top to flow the samples one at a time into the magnet. So because this is quite an efficient process, it's been possible to conduct a lot of research on hundreds of thousands of research samples. And these over the years have led to over 850 publications of NMR lipoprofile data. So it's really something sort of different than mass spec in that it's harder to generate the clinical association information that's needed prior to clinical translation from mass spec. It's easier by NMR. So we know an awful lot about the clinical value of what the NMR spectrum produces. So the intention all along actually was not to have a lab testing business, but to make these analyzers available to other laboratories. And to this end, FDA clearance was achieved for the first proprietary analyte, LDL particle number, and then the analyzer itself called Vantera. Millions of tests were performed, and several of these Vantera analyzers were actually distributed to reference labs. And so really what makes possible routine NMR testing is an instrument that looks nothing like a research NMR spectrometer. That's the whole idea. This is meant to be operated by a medical technologist with no training or knowledge about NMR whatsoever. And this, as I said, was FDA cleared. It really is successfully met the goal of being very simple to operate. There's no reagents, no consumables, so it's a very efficient and inexpensive test to perform. And you can see some of these other characteristics. The samples are introduced in the form of the original blood drawing tube, so there's no pre-analytical processing necessary whatsoever. The samples are withdrawn and diluted on board the instrument and flowed into the magnet, not introduced into the magnet in separate glass tubes. So that's about all the time I have to tell you about Vantera, but I do want to show you a 30-second or so video that was generated to show people at clinical chemistry meetings. So there you have it. But as I said, in 2014, the company was acquired by LabCorp, and their interest was in performing these tests in-house. And so these analyzers that were in these reference labs were brought back. And the main activity since then has been devoted to developing new LDT assays and clinical applications. So I'm going to share some of those with you in the last few minutes. So it turns out that the best clinical fit for NMR, as I've sort of indicated, is for high volume routine testing as opposed to esoteric tests, primarily because you're restricted to the fairly abundant metabolites that are in plasma. But since many of these are lipid related, it's not surprising that cardiometabolic disease is the sweet spot for NMR testing. And of course, cardiometabolic disease is something that is on people's minds post-COVID. In particular, this made people particularly susceptible to adverse consequences of COVID. But it's not just cardiovascular disease, it's diabetes, it's the obesity epidemic. And so all of this is a great concern and really a reason why we really need to do a better job with prevention. So this is a good review article that describes cardiometabolic-based chronic disease and how it's driven by obesity, adiposity. So you see adiposity at the top, you see the diseases, diabetes and cardiovascular disease at the bottom. But what actually causes the diabetes and the cardiovascular disease is not the adiposity per se, it's the intervening metabolic dysfunctions. And so this is, there's hyperglycemia associated with this and dyslipidemia, but the main drivers of the diseases are insulin resistance and inflammation. And the usual testing that gets done in a lipid panel, for example, does not include these very important contributors to cardiometabolic disease. So if one wants to do a better job with prevention, so-called precision prevention, as people have talked about, like precision medicine, what would make this possible would be a repurposed and upgraded lipid panel that would be performed instead of a regular lipid panel that just worries about dyslipidemia. So dyslipidemia and hyperglycemia already are being tested for, but insulin resistance and inflammation are not. And it just so happens that the single NMR blood scan not only gives you the dyslipidemia and the hyperglycemia, but it gives you, importantly, a good measure of insulin resistance and also inflammation. So I want to talk about those two elements now. First of all, insulin resistance. And something that I think people don't generally appreciate is that insulin resistance is the root cause of diabetes. It causes over time progressive loss of beta cell function. So the beta cells aren't able to produce enough insulin to overcome the insulin resistance. And so that leads over time to a loss of beta cell function that translates into hyperglycemia, which is mild at first and then gets greater, prediabetes, and finally diabetes. And as you probably know, what is happening these days is that diabetes prevention is really focused only on people that already have hyperglycemia, prediabetes. And that's primarily because it's easy to measure glucose and it's not easy to measure insulin resistance. So rather than focusing prevention efforts on the root cause of diabetes, the focus is on essentially the effect, the hyperglycemia. And in so doing, you're really losing the opportunity to intervene early and do something about diabetes risk. So it turns out that a single NMR spectrum produces 13 metabolites that combined give you a good readout of insulin resistance. This consists of the lipoprotein insulin resistance score that has 6 lipoprotein contributors to it, branched chain amino acids. And then since we haven't published on this NMR-IR score, I'm going to not disclose what these additional 4 metabolites are. But I just want to show you that in a large population of people that had this measurement performed, if you use the NMR-IR score to divide people into deciles, and then you look in each of these 10 subgroups at the insulin resistance as measured by insulin and fasting glucose, you see that in the red bars a gradually increasing insulin resistance associated with the higher NMR-IR score. But then you also see in the blue bars, with a 10-year follow-up period after the NMR measurement was performed, this is the rate at which future diabetes occurs in these 10 subgroups. And you can see that the NMR-IR score is definitely tracking with the prevalence of future diabetes. So this is really a quite useful biomarker. And then turning to glyc-A, the inflammation marker, I talked about its quantification earlier, but it is actually quite useful because the NMR signal is contributed to by several acute phase proteins. And so essentially how big this signal is a measure of systemic inflammation, a sort of a composite measure of inflammation. And because it's composite, it has generally stronger disease associations than CRP, which is the main inflammation biomarker that's used clinically. And it also has much lower biological variability than CRP, so it doesn't bounce around a lot from day to day. And just one quick example of a paper published last year that directly compared glyc and CRP for the prediction of future myocardial infarction and stroke and other cardiovascular outcomes. This shows that for myocardial infarction in particular, that glyc has a stronger ability to predict these events than CRP. So what is really quite remarkable is that a single inexpensive NMR scan produces all of the information shown here on the left. It could be output in the form of a patient report that might look something like this. The metabolites that together generate the insulin resistance marker, 13 in this case, is a good way of taking metabolomic information and making it, outputting it in simple form for clinical translation. And then finally, what I haven't mentioned yet is what's at the bottom, which is this additional multi-marker called MVX, or the Metabolic Vulnerability Index. And I'm going to spend the last couple minutes telling you about this marker of mortality risk, which is really quite remarkable. This is something that's brand new. This was reported on for the first time just 2 weeks ago in a paper in the Lancet Healthy Longevity. And the interpretation paragraph of the abstract says the following: that MVX made a dominant contribution to mortality prediction in patients with both cardiovascular disease and in low-risk subgroups without pre-existing disease, suggesting that metabolic malnutrition inflammation syndromes might have a more universal role in survival than previously thought. Obviously, you need to read the paper to understand exactly what all that means, but in terms of the ability of these markers to predict future mortality, I just want to show you data from the CathGen study. So this is about 6,000 patients that came to the doctor for cardiac problems, for cardiac catheterization, and over a 5-year period of time, 1,000 of them died. And if you generate a prediction model that has these 15 parameters, including age, and you look at the relative contribution of each of these to the overall prediction of mortality, you can see that these 6 parameters here make a very substantial contribution. These are 6 parameters that come from a single NMR scan. And when you brew these together into the MVX score, you have a score that contributes about 70% of the overall prediction of the model, that includes age and all sorts of other things that one would imagine are quite important predictors of mortality, like whether the patients have heart failure or diabetes or hypertension, et cetera. So this is really quite remarkable in terms of how dominant its prediction is of mortality, something that really was not known before. And this is a good example of how remarkably well these 6,000 people in this study were differentiated just on the basis of their MVX score at baseline. You can see that the numbers in parentheses are the number of people in each of these MVX categories. So if you have an MVX score less than 35, you have only about 3% of that group have died after 5 years. If you're unfortunately in the red group, then over 60% of the people in that subgroup have died. But the remarkable thing is that for even small differences in MVX score, there's a corresponding difference in the cumulative incidence of mortality over time. So this is really quite interesting and remarkable. And the clinical implications of this, I'll leave it to have you read the paper, but are quite profound, because it suggests that survival may depend more on previously unrecognized causal factors that are related to malnutrition and inflammation than those that are thought to actually be the causes of death. So this sort of cartoon indicates that the traditional thinking is that you die from things like cardiovascular disease, cancer, and of course older age. But the intervening metabolic vulnerabilities really dictate largely whether you succumb to any of these diseases. And so by at least recognizing this and having a biomarker that does a good job differentiating people who are metabolically more resilient versus vulnerable, as it says here, treating these underlying metabolic dysfunctions might provide greater survival benefit than targeting conventional disease risk factors. So it remains to be seen how useful in the future this will be, but it's just the beginning, but it's really a quite remarkable finding. So finishing up, I just want to sort of summarize the various things that one might expect would be necessary for routine NMR testing to be broadly clinically translated. And what I'm showing is that most of these have been accomplished. There are unique NMR biomarkers that address major chronic diseases. They've been extensively published on, they're extensively validated. There's an NMR analyzer that can produce this information very efficiently and cheaply. It's been used in this way for over 10 years. Regulatory challenges have been addressed. There are a broad international patent portfolio, and these assays and the technology is available for licensing. The only thing that's been missing and continues to be missing is an IVD company to basically deliver the analyzers and metabolic assays to clinical labs worldwide. But hopefully, with all of this background and 35 years of work that's gone into proving that this is a viable proposition and could be very useful clinically, hopefully an IVD company will be motivated to bring this to patients and clinicians. So with that, I just want to quickly acknowledge the very important contributions made by the scientists, engineers, and staff at LipoScience over these many years. None of this would have been possible without their work. So with that, I will stop and wait for the discussion.